Okay, I'm Rebecca. Hi guys, I'm Janine. And today we're going to be talking about the prognostic relevance of the mTOR pathway in renal cell carcinoma. But before we get to the paper, we just kind of want to take a step back and talk about cancer in general and the hallmarks of cancer. So the hallmarks of cancer is something that we've been talking about since pretty much the first day of class. So it was an article published in 2000 by Hanahan and Weinberg. And what they thought was that the complexity of cancer could be simplified down to a few key components, which are graphically illustrated here. And what we wanted to take the time to show you is how the PI3K, AKT, mTOR pathway relates to some of these hallmarks of cancer. So what the pathway does is it inhibits apoptosis. It also increases angiogenesis. It allows for sustained growth signaling. And then the last component is that it is involved in the switch from oxidative phosphorylation to aerobic glycolysis. So we just kind of wanted to point this out and how involved in many of the particular functions of cancer, how involved the PI3K, AKT, mTOR pathway is. And this is why this particular pathway is of interest to researchers to be able to target this pathway in order to combat cancer.
Yeah, and so taking you back to our chalk talk, this is the PI3K, AKT, mTOR pathway. It is very similar to the one that we drew on the board, but it includes different components. And so what I'd like to bring attention to are these three proteins that are included in the paper extensively, and their expression levels are what the research scientists are looking at in the renal cell carcinoma tissues.
All right, so just to kind of give you a little bit of a reminder of what we talked about in the chalk talk, because these three components are what the paper focuses on, we just wanted to kind of get you back up to speed. So AKT is also known as protein kinase B, and it contributes to three separate aspects of the hallmarks of cancer. So one of those is it inhibits apoptosis. A second one would be it increases GLUT1 levels, and we all know that GLUT1 allows for more glucose to be brought into the cell. So by having more glucose within the cell, that is helpful for the cancer cells, because we know that the cancer cells are heavily reliant on glycolysis. And then the third aspect of the hallmark of cancer that AKT is involved with is that it's the intermediate step in the activation of mTOR, which leads to subsequent upregulation of translation, which leads to the increased growth and proliferation that's associated with cancer. That's the first component.
Yes, and so P10 is another crucial component of this pathway. It is what regulates this pathway in normal tissue cells, and it is usually inhibited when in cancer cells. So usually in normal cells it acts as a tumor suppressor. Because as you can imagine, it's not in the pathway, but we mentioned it in the chalk talk. There are two other components not included, but in between PI3 kinase and AKT, and that's PIP2 being phosphorylated to PIP3. And that's what P10 is supposed to be inhibiting. And then, but what we see in cancer cells is that P10 is mutated with a loss of function. So that PI3 kinase is constantly activating AKT and consequently mTOR and its downstream effects.
So the final component of the pathway that they looked at was the S6 kinase. And what S6 kinase does is it leads to increased translation, which increases HIF1 alpha levels, which is something that Ryan and Claudia talk about, and it leads to the increased growth and proliferation associated with cancer. So it leads to sustained growth and survival. It is overexpressed in most tumor cell lines, which is something that we talked about in our chalk talk. And then something that we're gonna explain further, that the paper explains, is how this S6 kinase is the strongest predictor of survival in renal cell carcinoma. So that's something that the data is gonna show you throughout the paper. And since the paper focuses on renal cell carcinoma, we thought it would be a good idea to kind of explain some of the terminology that the paper uses. So there are two types of kidney cancer. There's clear cell and then there's non-clear cell. So clear cell makes up the bulk of the kidney cancer type. So it's seen in 75% are the clear cell type. And what clear cell is, it's usually a more aggressive form of the cancer. It also is the most receptive to this immunotherapy or targeted therapy that the end of the paper kind of looks at. And then non-clear cell renal cell carcinoma is kind of just like a broad category where they put in all the other types of renal cell cancer it is. So it involves the papillary cancer, which me and Tim talked about last class, and a bunch of other different types of cancer, or renal cell cancer. So what non-clear cell renal cell carcinoma is it's usually the rarer forms. Some of the subtypes are really slow growing, so they rarely ever spread. They are also not as susceptible as the clear cell to the targeted therapy that we're going to be looking at. So they're usually treated more so with chemotherapy. And then renal cell carcinoma, there's different stages. And in order to stage the renal cell carcinoma, they use the TNM staging system. And then if you read the paper, in a lot of the figures, it had like T classification, N classification, M classification. So we wanted to kind of just make sure we're all on the same page with those. So T classification relates to tumor size. So the higher the T classification, 1, 2, 3, or 4, the bigger the tumor is. And then N is whether or not the cancer is present in the lymph nodes. And then M is whether or not the cancer has metastasized to distant organs. So stage 1, it is associated with the best prognosis. The cancers are less than 7 centimeters and they are localized to the kidney. And then in stage 2, the tumors are greater than 7 centimeters but still are just localized to the kidney. Stage 3, the tumor has begun to grow and invade nearby blood supply, but it's seen in regional lymph nodes, but it has not metastasized yet. And then stage 4, which is associated with the worst prognosis, the tumor has grown exponentially. It has gone past this Geroda's fascia, and it is present in the lymph nodes and has metastasized to distant organs.
Yes, and so finally getting into the actual paper that we will be discussing today, the first couple of figures we'll be looking at expression levels of PAKT, P10, and PS6. And so what they did was they got biopsies from the patients. And so using the T classification, as Becca stated, the term kind of having a universal scale for tumor size. T4 means a greater tumor size, whereas T1 would mean a smaller tumor size. And then Furman grade is what determines the size and shape of the nucleus in the tumor. A G1 determines best prognosis, G4 is worst prognosis. And what the patient and tumor characteristics showed, they had 375 patients between 27 and 80 years old with previous nephrectomies. And what they did was they took their tissue biopsies, which were 0.6 millimeters in diameter and 0.4 micrometers in thickness. And what they did was they used immunohistostaining. And so this protocol is very similar to what they use in the paper, but has some deviation. But the idea is the same. What they would do is, with the tissues, they would put them on a slide or a plate. The different colors represent the different proteins present in the tissue. And what they did was they tagged the proteins with its specific primary antibody and then a secondary antibody to either show immunofluorescence, which isn't in this paper, or show the color of staining. And so in the first figures, we see these tissue samples. This is going to be normal tissue cells. What you see here is the purple is going to be the nucleus, and what we want to be focusing on is the brown coloration, which will be representative of PAKT and the other proteins that we're looking at. And so what we see in this paper is that in RCC, the expression of PAKT has increased, as you can see, with increased brown coloration. I would also like to bring attention to low grade versus high grade. Low grade means that the cancer is small and slow growing, whereas with high grade it's more aggressive. But as you can see, PAKT expression is increased in both. So it doesn't matter if it's low grade or high grade, you'll still see the increased expression. And so here we see the quantification of it. We see that cytoplasmic PAKT correlates with worse prognosis because it has a higher percentage of expression in T4 cells, which are the bigger sized tumors with the worst prognosis. It's also in metastatic and significantly more expression in clear cell, which, as Becca mentioned before, is correlated with the worst prognosis. However, the scientists also looked at nuclear PAKT, and they found the complete opposite correlation, that it had an increased expression in T1, which is associated with best prognosis, with localized tumor cells, and the clear cell versus non-clear cell difference isn't very significant. Now looking at P10, this may be a little bit confusing because with PAKT in cancer it's increased, but with P10 it's decreased. And so in normal tissue cells we would see a high expression of P10, as you can see here. But with cancer, it has a decreased expression in low-grade and high-grade RCC. And we also see that quantified. We see a higher expression of P10 in the best prognosis of tumors in T1. We also see it in localized and in non-clear cell, all associated with the best prognosis. And finally, this is the tissue stain for PS6. And as you can see in normal cancer tissue, it has a very low expression, but in RCC, it has increased expression. And again, this is significantly across the board that it has a significant increase in expression in T4 tumor cells in metastatic and in clear cell tissues.
All right, so the second point that the paper brought up was how these expression levels that Janine just looked at, how those relate to survival rates. So Janine talked a little bit about how the difference between the cytoplasmic AKT and the nuclear PAKT, and if you, as she was saying, the cytoplasmic PAKT, when it's seen in high levels, it's in stuff that we associate with a poor prognosis, so large tumors that have metastasized. So it makes sense if you look at the gray line. These legends are kind of weird. I'll point them out every time. But you want to look at the gray line means for high levels of cytoplasmic PAKT. If you look at that, you get a worse prognosis, or lesser survival rate, which would be what you would expect because this is associated with the things that are associated with a lesser prognosis. And then the complete opposite is shown for if you look at the nuclear PAKT. So the black line actually represents higher levels of the nuclear PAKT. And that was, the nuclear PAKT was expressed greatly in things that we would associate with a better prognosis, so small tumors that were localized. So high levels of nuclear PAKT have better survival rates. And then the next one is looking at P10. Again, we talked about how high levels of P10 would be associated with a better prognosis, and that's just seen here. The higher levels of P10, so above 75%, that you see with the black line, led to an increase in survival rate. And this makes sense if you think about it because, as we were talking about earlier, P10 is a tumor suppressor. So if you have high levels of P10, you are not going to have that phosphorylation of PIP2 to PIP3. So you're not going to get the activation of AKT, not going to have the activation of mTOR. So then you won't see the increased translation, the growth and proliferation. So if you have higher levels of P10, it makes sense that you're going to have a better survival rate. And then the last thing that they looked at was how lower levels of S6 or PS6 lead to increased survival rates. So again, this time you want to look at low levels would be the black line. So you'd see the increased survival rate. And what I really just kind of wanted to point out here was how big of the gap is between this. And that's just in comparison to the P10 and the PAKT levels, where the expression levels didn't make too much of a difference. This gap is much greater. And they pointed out in the paper how it is the PS6 that is the best indicator of survival rate. So if you see high levels, you're going to have a much higher survival rate. Or if you see low levels, you're going to have a much higher survival rate than if you see high levels. And then finally, the paper pointed out how those survival rates that we just looked at, how those are correlated to the localization of cancer, so whether it's localized or it's been metastasized. So this is how I like to explain it. So if you have a localized renal cell carcinoma, your best chance, in order to get the best rates of survival, you would want to have high nuclear AKT expression, which is shown by that black line, and you would want to have low PS6 expression, also shown by the black line. And that just makes sense if you think, this kind of graph just kind of combines all the graphs 2 and 3 that Janine looked at, and then the graph 4 that I looked at. And then again, if you do happen to have the metastatic version of the renal cell carcinoma, your best survival rates are going to be seen when you have low cytoplasmic PAKT as well as low PS6 levels and then high P10 levels. So these graphs just kind of combine everything that the paper has been talking about. And then just, the paper brought up some future directions for therapy. So what they looked at, they looked particularly at 3 esters of rapamycin, which were 10-serolimus, AP23573, and RAD001. And what these do is they directly inhibit mTOR, so you're not going to see the increased translation and growth and proliferation. But they actually do not act by, they don't cause apoptosis in the cell. What they actually do is they induce G1 growth arrest. So they're not going to kill the tumor that's already there, but they're just going to stop it from growing any further. So if you guys remember from our chalk talk when Janine was specifically talking about using combination therapies with this type of pathway, because you could use any of these rapalogs and you could target mTOR, and you can shut down the pathway so it's not going to have further proliferation, but you would want to pair it with something that would induce apoptosis in order to see the best results. And when these rapalogs are going to work best are in patients that have highly active mTOR, and the best way to kind of determine whether or not a patient has a high active mTOR is to look at the S6 level. So what you would do is you take a biopsy of their tumor, do one of those immunohisto stains, and if you see the high levels of the S6, you would want, those patients would be ideal for using this type of treatment of the rapalogs combined with something that would induce apoptosis.
And so, okay, bringing it back to the pathway and to the main points of this paper. So we see PI3 kinase, AKT, mTOR, PS6. They're all very important components with cancer and with cell proliferation. And what this paper showed that the expression levels of P10, AKT, and PS6 correlate with either worst prognosis or best prognosis, depending on whether or not their expression levels are low or high. And also survival rates are very important correlation with these expression levels. And so thinking about it in the greater scheme of things, if scientists see this in renal cell carcinoma, they have also seen it in breast cancer. If they look at PS6 expression in cancers that have high mTOR activity, then this treatment with the mTOR rapalogs would be very beneficial for patients in the future. Questions?
I met him yesterday.
Janine added the coffee.
Yeah, I forgot about that. I don't have
Okay, any questions? No? Okay. So some of the results that you told us came from different tests that were performed. So the Kruskal-Wallis test and the Mayo-Whitney test. Can you guys explain any part of it or just what that even means? Or even if you don't know the specifics of that test, just what are they used for, like in general terms? Like what data corresponds to those tests?
Is that the graphs? I'm gonna say it's probably the graphs. We didn't really look at these specific protocols for those tests. We were more focused on trying to interpret the data.
What I think it was like that they were using the different like T classification things and they were applying it to those, I don't even remember what the term was, but those tests. And those tests are ways in which that they can make it like, they're like uniform tests so that everything with, so like if you're at one hospital and they're looking at your tumor, they're gonna have the same results as if, it's like ways of, I can't think of the word, universalizing like the way that they look at cancer.
Yeah, it was all the patient-based stuff. They're usually very data heavy, like how you can analyze patient samples and actually put them in groups and compare them to one another. So it got brought up by a couple people, so I just wanted to see if you if you guys had read anything about that. So while we're on the whole part about the patient part of it, patient test. So this is all done retrospectively. So do you think that had any possibility of a bias of what samples were seen here, or does that not matter?
I'm sure there's some sort of underlying bias with with results presented in the paper, they did have 375 patients, and I don't think that in the paper they mentioned if these tissue samples were coming from a smaller pool from those patients, or if they included data from all of the patients altogether, or if it was just quantified out of all the 375 patients. So there may have been a little bit of bias, but as you can see, if we go back, the expression levels are significant.
And then one last question, which I'm sure you guys thought about too, is this nuclear versus cytoplasmic difference in phospho-AKT. Do you have any hypotheses for why cytoplasmic is so bad and nuclear?
We were looking at that, and we found that both nuclear and cytoplasmic is involved in regulating the cell cycle. And so, thinking about it, we focused on like nuclear AKT probably is more involved with DNA synthesis and specific parts of the cell cycle, whereas with cytoplasmic AKT can also be involved in phosphorylation kinases and activating other proteins that may result in increased translation.
And I guess one last question, maybe if you go back a few slides, because you had one that was like a summary slide that I think showed them all of the different survival graphs, but you had put like all three to get that one right there. Yeah, so if you look at like when they looked at different proteins, their cutoffs, so the percentages of what they were viewing, like higher or lower, is 35, 35, and then for people, S6 is like 70. Is there any comments in the paper or any hypothesis of why like their limits were so different when they were looking at test subjects for different groups?
I think they were, I do not remember.
I don't think these might not have been said, and do you just have an idea why, like, if you logically were thinking about it, why one protein you'd be looking at differences between like 80% or like 60% versus the other, you're looking at like 80 versus 20?
Maybe because PS6 is more downstream in the mTOR pathway, and it had more significant expression level, and so PS6 is a more crucial component because it's downstream of mTOR.
Just how sensitive kind of proteins are to activation. Okay, good job, guys.